Just a little bit of background about my team and the Genome Canada Transplant Consortium. It was established back in 2015, and our program links investigators, healthcare providers, patients, and policymakers across North America, Canada, and Europe, as well as some other nations, including Australia and in Asia, in the fields of autoimmunity, transplant, tumor immunity, and infectious immunity. I focus my research in the area of transplant immunity, but as you can see from this diagram in one of our publications looking at precision medicine applications in transplantation, really immune health is the nexus of all of these areas. And in fact, in transplantation, all of these are also relevant in the immune context. So the Genome Canada Transplant Consortium has been very fortunate, and they've been funded for a number of large-scale programs, starting in 2017 with the CAN-PREV program. And one of the key pieces of that grant was actually nanopore sequencing of HLA genotyping for transplantation. That ended actually this year, and we were also successful in following up that program with a clinical trial called CAN-PROSPECT, which runs for the next 3 years, which is underpinned by the application of rapid sequencing of HLA genes and using that information for the definition of HLA epitopes in order to allocate organs between patients and donors that come into our laboratory. And all of this is actually building a foundation for the end goal of our program, which really is to bring operational tolerance to reality. Operational tolerance is this hypothesis that we could get our patients either totally off immunosuppression, or at least to a minimal dose of immunosuppression whilst they have their graft in situ. This would be great for a number of reasons. Immunosuppressive drugs have a lot of side effects, and those immunosuppressive drugs not only cause neurotoxicity, but also increased risk of developing viral infections, as well as certain types of conditions, including post-transplant lymphoproliferative disorders. And so being able to get to the point where we could provide tests, diagnostic tests, biomarker assays, and understanding the immune mechanisms that underpin operational tolerance would really benefit our patients and maintaining their graft and the health of their immune system through the rest of their lives. The Canadian Blood Services is a national program that oversees the provincial healthcare providers that provide transplant services in each of the provinces in Canada. It also runs the national programs, which include programs where we share organs across different provinces, and they provide support services for knowledge translation and strategic planning. One of the reasons why I wanted to bring this up is because as an output of one of our first programs, the CAN-PREV program, we were successfully able to translate nanopore technologies into each of the 14 HLA laboratories across Canada in collaboration with Canadian Blood Services, making us the first country that is nanopore-enabled for transplant diagnostics in the HLA space. Outside of HLA for transplant, we also test HLA for adverse drug reactions. And actually, this paper has just gone in for a review. We were able to do a proof of concept using nanopore sequencing for precision prescribing. Many of our patients have particular HLA genotypes that are risk alleles for developing adverse drug reactions. And at the moment, the standard of care test that we use has a 2-week turnaround time, by which point the patient's gone home, doctor's already had the prescription written for the patient, and it really isn't a useful test in order to help patient management. And so what we did was using the same protocol in terms of prescription and clinical sample collection, we used nanopore to do the HLA genotyping on the risk alleles for these patients and managed to get results back to the physician in 4 hours, at which point the patient's probably still in the clinic. And they're able to either assign an alternative therapy if the risk alleles are present in the patient. So this is just one of multiple ways that nanopore is already changing our clinical practice in Vancouver. But I wanted to talk about adaptive sampling and how that is one of the approaches that we're using for the future of getting us to operational tolerance. So what we wanted to do was develop a comprehensive risk assessment for immune risk in transplantation, an all-in-one test, as you will, say. So a complete sequencing of HLA genotyping provides more information. We also want to assess non-HLA immunogenic loci, which may also be involved in the modulation of immune responses. Sequencing of PGX genes, we've heard a couple of talks today, we're talking about PGX genes, and also sequencing of ABO and minor blood group genes, all in one simple test. So a lot of this data comes from my colleague, Sarah Grazidiak, and she was able to do a number of optimizing strategies on the nanopore assay perspective only. The reason is because I wanted to maintain obtaining and processing clinical samples as standard of care clinical samples. There is no changing in the DNA extraction method, no changing required at all at that point, and only the sequencing portion that was optimized. We included 164 regions of interest, including the complete HLA locus and some other non-HLA pharmacogenomic locus as well. So here, I just wanted to show an example. This was run on a PromethION flow cell. We did shear the DNA to approximately 15 kb. It was overloaded, and it was a 20-hour run. And you can see really nice enrichment and about a 10x low-pass genome in the background there. And this is just one example of the regions of interest that we used, and this was in a 2-day turnaround time, but this essentially replaced about 10 to 15 clinical tests. So the initial plan for us was to evaluate HLA, blood group genes, and PGX genes all in one, and we are currently able to infer haplotypes for HLA, and we've just used a couple of different aligners there, providing 2-field or 4-field resolution for those genes. We also were able to infer the typings for 46 of the ISBT blood group genes and 20 CPIC level PGX alleles as well. And we also run the variant callers, and a lot of other steps are already underway. The key thing that I wanted to mention was that this pipeline is exceedingly flexible, and Sebastian actually alluded to this in his talk. There was this example where a physician emailed me and asked if we had a genetic target that I could run for him. My postdoc actually saw the email over my shoulder, and then the next day had changed the bed file and given me the data straight away. Like, it's in 24 hours from an email to a result is just unbelievable. So I just wanted to say thank you for all the members of the BC Immunology Lab, our VCH collaborators, and the Genome Transplant Consortium, and appreciation for all their contribution and their support. Special thanks to the Nanopore teams here and in Canada who have been wonderful in helping us getting this set up. And of course, recognition to all our patients and their families for their contributions.